Hello and welcome back to Study Hub Pro. Today we're going to do something pretty amazing. We're going to crack the code of life itself. We're going to figure out how we can actually predict the traits that get passed down from one generation to the next.
I mean, that's the big question, right? How can we know what's coming? Because genetics isn't just looking backwards at what's already happened. No, it's an incredibly powerful tool for prediction. So come on, let's build our toolkit and get ready to solve this puzzle.
Okay, first things first, let's nail down the absolute basics. So a gene, think of it as a unit of inheritance, a piece of DNA that gives the instructions for a specific trait, like, say, hair length. And then you have alleles. An allele is just a different version of that gene. So you might have the allele for short hair or the one for long hair. All right, two other terms that are absolutely critical. First is genotype. That's the actual genetic makeup, the combination of alleles an organism has. It's the code. So if the alleles are the same, like big S, or little s, we call that homozygous. And if they're different, like big S, that's heterozygous. Then you've got the phenotype. That's the physical thing you can actually see, like short hair. And that's determined by which allele is dominant, the one that gets expressed, and which one is recessive.
Okay, you ready? Let's dive right in. We're going to start simple, tracking just a single trait. In the world of genetics, we call this a monohybrid cross.
So think of this as your four-step recipe for solving pretty much any one-trait genetic puzzle. It's simple. First, you identify the parents' genotypes. Then you figure out the possible gametes they can pass on. And from there, you can map out the first and second generations of offspring. Let's see how it works in action. So here we go. We're crossing a homozygous short-haired cat, that's big S, with a long-haired cat, which is little s. And what's the result? Well, would you look at that? Every single kitten in this first generation, the F1 generation, has the heterozygous genotype big S, little s. And because that short hair allele is dominant, they all have a short-haired phenotype. Wild, right? They're all carrying the code for long hair, but you'd never know it just by looking at them.
Okay, let's put this to the test. Time for a little genetic detective work. We've got 2 cats, they have 5 kittens, 2 short-haired and 3 long-haired. Now we know one parent is long-haired with the genotype little s. The question is, what's the other parent? The huge clue here is those long-haired kittens. Since long hair is recessive, their genotype has to be little s. And that means they absolutely had to get one of those S alleles from each parent.
So follow the logic here. We know one parent is SS, which means it can only pass on a little S allele. So for any kitten to end up as SS, the other parent must have a little S to give, right? But wait, we also have short-haired kittens. That means that same parent also has to carry the dominant big S allele. So what's the only genotype that fits? It's gotta be heterozygous, big S, little s. A short-haired cat that's secretly carrying the long hair trait. Case closed. All right, we've pretty much mastered single traits, so let's level up. This is where things get really interesting. When we start tracking 2 or maybe even 3 traits at the exact same time. Okay, so when you cross 2 individuals that are heterozygous for 2 different traits, think Mendel's pea plants, looking at both height and color, you always get this classic 9:3 to 3:1 ratio in what you see, the phenotypes. This predictable pattern was a huge breakthrough. It proved that different traits are inherited independently. But what happens when we throw a third trait into the mix? Well, things get a little crazy. The Punnett square you'd need would suddenly explode to this size, 64 boxes. I mean, come on, that's not just tedious, it's a minefield for making a simple mistake. But thankfully, there is a much smarter way. We absolutely do not have to draw that giant chart. Instead, we can use the power of simple probability to get the exact same answer way faster and with a whole lot less risk of messing up. And this is it, the forked line method, or just the probability shortcut. Instead of one giant, scary problem, you just break it down into 3 small, totally manageable ones. You figure out the probability for each trait on its own, and then, and this is the beautiful part, you just multiply those individual probabilities together. Okay, let's try it out with our next practice question. We're doing a trihybrid cross. Both parents are big A, big B, big C. What is the probability of getting an offspring that's homozygous recessive for all 3 traits, so A, B, B, C, C? And this is where the magic happens. Look, for each trait, A times A, the chance of getting A is 1 in 4. Same for B getting B, and C getting C. So with our shortcut, we just multiply them. 1/4 times 1/4 times 1/4, which equals 1/64.
See? So much more elegant than wrestling with the 64-box grid, isn't it?
Okay, so everything we've looked at so far has been based on this idea that genes play by the rules and pretty much act on their own. But now we get to the really wild stuff. We're entering the secret levels of genetics, where genes stop minding their own business and start interacting with each other in really surprising ways.
And the two main rule breakers we need to talk about are epistasis and pleiotropy. Now, epistasis is when one gene can literally mask or modify the effect of a completely different gene. Think of it like a master switch. It doesn't matter if the gene for your light bulb says on if the master switch for the whole house is turned off. Then there's pleiotropy. This is where one single gene influences multiple, totally unrelated traits. It's like a domino effect.
Let's dig into epistasis a bit more, because it really messes with our expected ratios. That classic 9-3-3-1, epistasis throws it right out the window. A perfect example is coat color in mice. You've got one gene that determines the color, say a goody or black. But there's a second gene that acts like a pigment switch. If a mouse gets the homozygous recessive CC genotype for that switch, it's albino. It doesn't matter what the color gene says. The pigment factory is closed. And this completely changes the ratio to 9 agouti, 3 black, and 4 albino.
Okay, so that was recessive epistasis. Now let's try a question about dominant epistasis. In squash, a dominant W allele for white color is epistatic. It completely masks the gene for yellow or green. So if you get a 12-3-1 ratio, what do the different phenotypes look like? Here's how it breaks down. That dominant W allele is like a trump card. If a squash has even one copy of it, it's gonna be white. Game over for the other color gene. And that accounts for a whopping 12 out of the 16 possibilities. The only way you can get another color is if the squash is homozygous recessive WW. Only then can the second gene show its stuff. If it's WW and has a dominant Y, it's yellow. And if it's recessive for both genes, WWYY, then it's green.
All right, let's switch gears from one gene masking another to pleiotropy. And this quote really nails it. It's like a package deal of symptoms from a single gene. This is what happens when one single, faulty gene doesn't just cause one problem, but sets off a whole chain reaction of issues all over the body. And the absolute classic example of this is sickle cell anemia. It all boils down to a mutation in just one gene. The main effect is that it creates defective hemoglobin. This causes red blood cells to twist into this crescent or sickle shape. But from that one little change, you get this devastating package deal of symptoms. Those sickled cells clog up blood vessels, leading to terrible pain, rheumatism, and even organ damage. But, and this is the fascinating part, there's a twist. Having just one copy of this sickle cell gene actually gives you a high resistance to malaria. It's just a stunning example of how one gene can be a multitasker, causing both terrible problems and a major survival advantage. So we've gone from simple Punnett squares all the way to these incredibly intricate gene interactions. So, what's the big picture here? Why does any of this matter? Well, it matters because whether we're talking about a simple one-trait cross or a complex interaction like pleiotropy, these patterns, these are the fundamental rules that govern the code of life. Understanding them allows us to predict inheritance, to get a handle on genetic diseases, and to see evolution happening right in front of our eyes. This is the framework for so much of biology today, and it really just leaves you with one big question, doesn't it? What other secrets are still hidden away in our DNA, just waiting for us to find them?